infectious diseases, research, medicine, health. Welcome to Outbreak News Interviews. And now, broadcasting from the Outbreak News Skylar Studios in beautiful West Central Florida, here is your host, microbiologist and editor of OutbreakNewsToday.com, Robert Harriman.
Well, hey everybody, this is Robert and welcome to today's show. Now, a vaccine against the bacterium, Burkholderia pseudomallei, that causes melioidosis, was tested in a mouse study and found to be highly protective against the disease, according to UCLA researchers. So what is Burkholderia pseudomallei? What is melioidosis? And what about this vaccine study? Well, joining me today to answer these questions and more is Marcus Horwitz, MD. Dr. Horwitz is a distinguished professor of medicine in the Division of Infectious Diseases and of Microbiology, Immunology and Molecular Genetics at the David Geffen School of Medicine at UCLA. He is also the senior author of the paper on this vaccine study in the journal mBio. Dr. Horwitz, welcome to the show, sir.
Thank you. Nice to be on.
Okay, well, before we get into the vaccine and the study, let's give the audience a little bit of background on this pathogenic organism and the disease it causes. Can you talk a little bit about Burkholderia pseudomallei and melioidosis, which are not exactly everyday terms that most people are going to know about?
Sure. Yeah. So this pathogen is a bacterium, and it's what makes it unusual, it's in a class of pathogens that are called intracellular pathogens, meaning they have the ability to invade and multiply inside white blood cells. As you know, white blood cells usually defend us against microorganisms, but in this case, this select group of pathogens has developed the ability to frustrate our host defenses and to use the white blood cell actually as a host in which to multiply. And that's why it's in a class of pathogens that are fairly difficult to protect against, and they cause serious diseases such as melioidosis, but also diseases like tuberculosis and leprosy and tularemia and so forth. Now, as for the disease melioidosis, this disease can manifest itself in several ways. The most frequent manifestation is a serious pneumonia, but it can also infect the skin and cause ulcers and abscesses. It can infect the genitourinary tract, prostatitis, urinary tract infections, kidney infections. It can infect bones, joints, even the brain, and cause encephalomyelitis. And the pneumonia that it causes can be akin to tuberculosis, and in fact, is often misdiagnosed as tuberculosis. And the organism does share several features with tuberculosis, such as being an intracellular pathogen, as I mentioned.
So where is the bacteria found geographically and environmentally, or?
Sure. So this bacterium, it's found worldwide, but it tends to be found abundantly in areas of the world that are tropical or subtropical. So most of the cases that are reported, and by the way, about 165,000 cases occur each year and about 89,000 deaths. So this is a serious major disease, not in the United States where it's fairly rare, but in these other areas of the world. So most disease comes from Southeast Asia, South Asia, India, China, Northern Australia, and most infections are reported from Thailand, Malaysia, Singapore, and Australia. But it also causes disease in other tropical and subtropical areas, for example, in Africa or even in the Americas, including, incidentally, Puerto Rico and the Gulf Coast of the United States.
Right. So how does somebody contract it?
So in the endemic areas, people will typically contract it by skin inoculation from contaminated soil and contaminated water, especially during the wet season. But it can also be contracted by inhalation. particularly during the monsoon season or during severe weather events, which will stir up wet soil and so forth. And finally, it can be contracted by ingestion of contaminated water, and that's one of the ways that it's felt that children are able to get parotitis, which is infection of the parotid gland from drinking contaminated water.
And it is a bacterium, so I assume it's treatable with antibiotics.
Yes.
However,
Well, there's good and bad news there.
yeah.
The good news is that, yes, you can treat it with certain antibiotics, but the organism is resistant to the major antibiotics that we are used to treating bacterial infections with. So it's resistant to penicillin, ampicillin, aminoglycosides like gentamicin and kanamycin, streptomycin, and so forth. But fortunately, there are some new generation, third generation, fourth generation cephalosporins, meropenem. There are some new drugs that can treat it. But there's also other bad news, which is that unlike most bacterial infections, this one requires a very prolonged treatment. And in fact, treatment is usually in two phases. There's what they call the intensive phase, where the patient is treated with intravenous antibiotics for about 2 weeks. And if that's successful, then one goes on to what they call the eradication phase, where the patient is treated for 3 to 6 months with oral antibiotics, because this infection can cause abscesses throughout the body, and these can be fairly difficult to clear.
Yeah, and I imagine untreated, the prognosis is not very good, right?
It's not good, actually. Even in a resource wealthy country like Australia, even there the fatality rate is around 10%, but in resource poor countries, which is where most of the cases occur, the fatality rate's about 40%. Yeah. So it's a fairly fatal disease.
Yeah. Now, it's also classified as a tier 1 select agent of bioterrorism. So this is the same category as anthrax and plague. Can you just explain to the audience briefly what that means?
Sure. So the tier 1 select agents are a group of pathogens that are considered to be most likely to be used in a bioterrorism attack. And the reason for that is because these particular agents can cause highly fatal disease by exposure, airborne exposure, at fairly low doses. And so it includes diseases like tularemia, plague, melioidosis, the agents that cause those. And Burkholderia pseudomallei, and as well as its sister organism, Burkholderia melii, are in that category of select agents.
Yeah. Let's go ahead and switch gears to the vaccine and the study that you were a major part of.
Okay.
What kind of vaccine is it?
Okay, so this vaccine is comprised of what we call bacterial vector, which is just a bacterium that expresses proteins, in this case, those of Burkholderia pseudomallei, that are able to induce an immune response that can protect the host from disease. Now, the vector is something we've used as a platform for other vaccines, including for other tier 1 select agents like tularemia, anthrax, plague. This vector is actually derived from another vaccine that's already been used in humans. That's a tularemia vaccine called live vaccine strain, or LVS. Not a very imaginative name, but that's what they called it. This vaccine was developed in the 1900s in the Soviet Union, later gifted to the United States, and it's been given to over 60 million Russians, and the U.S. version to over 5,000 U.S. lab workers. However, the vaccine still retains a certain amount of toxicity. So to make it less toxic, or hopefully non-toxic, we attenuated it further. So this vaccine was attenuated originally by the Soviets, kind of the old-fashioned way, which is by passaging an organism on artificial medium. And in the process, it dropped 2 major genes, which are called major attenuating deletions, is how they're referred to, and several minor ones. What we did is, using more modern techniques of recombinant molecular biology, we knocked out another major gene that really attenuated it much further. In fact, it's greater than 10,000-fold less virulent in a very sensitive model. So then we took this vaccine and used it as a vector, the more attenuated vaccine, used it as a vector to express recombinant proteins of Burkholderia pseudomallei. We picked out several proteins and made a couple, what we call 3 antigen vaccines. So they're expressing 3 different proteins. And that's what the vaccine consisted of.
Yeah. So it's pretty, melioidosis is fairly uncommon compared to a lot of diseases. So
uncommon in the United States,
correct.
But it's a major disease when you consider 165,000 cases, 89,000 deaths a year. It puts it in up there. Yeah.
But why is this vaccine important? Who's the audience or the target?
Okay, so I would say it's important for 3 reasons. First, there is no licensed vaccine for melioidosis, number 1. So that means this vaccine could be used to reduce the risk of disease and death in the endemic areas of the world, such as Southeast Asia, South Asia, Australia, and so forth, where there are a high number of cases.
Right.
Secondly, the vaccine could be used to protect people who travel to these endemic areas, including military personnel, as well as tourists. And military personnel are particularly vulnerable because they tend to be located and engage in activities that come in contact with soil and water that's contaminated. They're more likely to be exposed. And finally, as we touched upon, it could be used to protect against an intentional bioterrorist attack.
Yeah. All right. So can you go ahead and talk about the results, the methods, the results of the study that you did?
Sure. So we tested this vaccine in an animal model, of course, because that's what one does preclinically.
Right.
And we tested this vaccine under highly stringent conditions. First, we selected an animal model, a strain of mouse that's unusually highly sensitive to this disease. Secondly, we selected a highly virulent strain of Burkholderia pseudomallei. And thirdly, we challenged these highly sensitive mice with this highly virulent strain of Burkholderia pseudomallei by the respiratory route. That's the route that's hardest to protect against.
Right.
And so this was a high bar for testing the vaccine. And what we found was that in the unimmunized animals, 100% of them died within a couple, 3-4 days from challenge with this strain of Burkholderia pseudomallei. In contrast, the immunized animals were highly protected against both death and disease. Now, we also, I should mention, tested both administration of the vaccine by the intradermal route and also by the intranasal route. And what we found was that both protected, but the intranasal route was better at protecting against very high doses of Burkholderia pseudomallei. And we found that even one immunization was able to give very high level protection. And that protection was also long lasting. We tested it over a longer period than usual, and it was a long lasting protection.
So what's next? Is this a vaccine that can be tested on human beings at some point?
Yes. So with respect to the airborne acquisition of Burkholderia pseudomallei, which is of greatest concern, the next step would be to test this vaccine in a second animal model. That's a term the FDA uses, the Food and Drug Administration. They require testing in a second animal model that's quite different from the first one to see that to make sure it's safe and effective in that model. And so that's our next plan. And then if that is successful, then the vaccine can go into humans for testing for safety and immunogenicity. It cannot be tested for efficacy, and that's why the FDA has this second animal model. They call it the animal rule, because it's for situations where it's not feasible to test a vaccine easily otherwise.
Yeah. That makes sense.
Yeah, yeah.
All right. Well,
let me just also mention we have other
please.
plans too, which is we plan to test. So this was by the most dangerous route, inhalation. We also plan to test the vaccine for efficacy against subcutaneous challenge, which is the way that most natural disease is acquired. And finally, we're also going to test it against Burkholderia mallei, which is a sister select agent to Burkholderia pseudomallei, very similar from same genus. And we suspect that we hypothesize it will work because it shares the proteins in the vaccine are shared largely by these two pathogens. So those are what our next steps are going to be.
Yeah, and B. mallei is the cause of glanders. Am I right?
That's correct. Glanders in humans and in animals. Correct.
Yeah. Okay, great stuff. You're doing some really good work over there in UCLA, that's for sure. And I'll go ahead and link to the study published in mBio in the show notes when I go ahead and publish the podcast for listeners that are interested in checking it out. And I want to thank you, Dr. Marcus Horwitz, for sharing your time and your expertise with us today. Thank you, sir.
Thank you for your interest.